不是这样的哈，因为我必须得听到他那边，我们还有一个对接，并不是我马上就开始，那边先开始我才开始，所以你屏幕上要有他们。我要看到那边的东西才行。
Okay, good evening and welcome to our webinar on the SDG-aligned Artemia aquaculture. I'm Simon Wilkinson from the network of aquaculture centres in Asia Pacific. Tonight's webinar is the first event of the Global Conference on Aquaculture Millennium Plus 20. The main conference will kick off tomorrow via Zoom, and I encourage you to register for that through the conference website. We'll provide a link at the end. Now we will begin the proceedings. I'd like to hand over to Professor Li-Ying Sui to introduce our first speakers. Professor Li-Ying, please go ahead.
Thank you very much, Simon. Dear participants, colleagues, and friends, good morning, good afternoon, and very good evening. Welcome you to SDG-aligned Artemia workshop. My name is Sui Li-Ying. I'm from Asian Regional Artemia Reference Center, Tianjin University of Science and Technology. First of all, I would like to introduce our distinguished guest at the head table. Professor Jiang Min, the Vice Rector of Shanghai Ocean University. Mr. Jia Jiansan, the former Deputy Director of Fishery and Aquaculture Division of FAO. Professor Wang Qingyin, the Board Chairman of China Society of Fisheries. Welcome. I would like to also to introduce my colleague, the co-moderator of the workshop, Dr. Rodrigo Rubach from FAO Rome. Rodrigo, hello, good afternoon.
Good afternoon. Thank you.
So now I'm honored to invite Professor Jiang Min to give a welcome address. Professor Jiang Min, it's your floor.
Distinguished Matthias, Jia Jiansan, Rodrigo Rubach, Wang Qingyin, and Sui Li-Ying, dear Professor Patrick, distinguished scholars and guests, good morning, good afternoon, and good evening. Welcome to the pre-conference workshop of GCA, SDG-aligned Artemia aquaculture. My name is Jiang Min. I'm the Vice President of Shanghai Ocean University. On behalf of Professor Wan Rong, President of Shanghai Ocean University, I would like to extend my heartfelt welcome to all the participants in the workshop and express my sincere greetings to all the experts and scholars. My heartfelt thanks also go to the staff who provided support and help for the meeting. Firstly, I'd like to say that it is our great honor to be one of the hosts of the Global Conference on Aquaculture Millennium Plus 20. As one of the oldest fisheries universities in China, Shanghai Ocean University, founded in 1912, will celebrate its 110th anniversary next year. And as early as 1921, it became the first educational institution in China to establish agriculture department and generated a group of famous scholars such as Zhu Yuanding, Meng Qingwen, and Li Sifa, who contributed valuable knowledge and experience to the aquaculture. Shanghai Ocean University is the one with the most complete specialty of higher education in the field of aquaculture in China, including 4 undergraduate majors in aquaculture. And also, we have the most comprehensive education system from undergraduate master, doctor to postdoctoral programs, cultivated many talents in agriculture at home and abroad. As we all know, Artemia is a small crustacean widely distributed over the world. Since Seal first used newly hatched Artemia nauplii as bait for juvenile fish in 1933, Artemia has been widely used in agriculture. Our Dr. Patrick has made outstanding contributions to the promotion and application technology development of Artemia. The Artemia Reference Center of Ghent University in Belgium, initiated by him, is still an important platform for Artemia research around the world. FAO, NACA, and other organizations such as Great Salt Lake Brine Shrimp Cooperative USA, such as Artemia Association of China, Asia Regional Artemia Reference Center in Tianjin University of Science and Technology, have made outstanding contributions to the promotion and application technology development of Artemia. However, further study and studies are needed to assure the sustainable provision of Artemia. That's why we are here together. All participants are encouraged to strengthen communication and cooperation in the future and jointly discuss the topic for SDG-aligned Artemia. Finally, I wish this workshop a complete success. Thank you.
Thank you very much, Rector Jiang. In fact, as the local facilitator, Shanghai Ocean University have made a tremendous effort for this workshop. We appreciate so much for your strong support for this workshop. And next, I'm honored to invite Professor Wang Qingyin from China Society of Fisheries to give a welcome speech. Professor Wang, please.
Dear Professor Jiang Mi, Professor Patrick Sokolos, Mr. Jia Jiansan, Mr. Simon, and Professor Sui Liying, colleagues and friends here in Shanghai, and all of you attending the video conferencing. First of all, on behalf of China Society of Fisheries, I would like to extend my heartfelt congratulations to the SDG-aligned Artemia workshop. Brine shrimp Artemia is a crucial livelihood for marine fish and crustacean culture. With the expansion of world agriculture, the demand for Artemia cysts have continued to increase. Today, the workshop is held at the pre-meeting of the Global Conference of Agriculture, fully demonstrating the importance of Artemia in fulfilling the SDGs for the agriculture industry. I am very pleased that this workshop is held in China, the country with the biggest resources of Artemia, from inland salt lakes and coast salt works, and the biggest consumer of Artemia in agriculture hatcheries for fish, shrimp, prawn, and crab. Except for the cyst product, Artemia are also sold in the form of newly hatched nauplii, fresh and frozen biomass, as well as decapsulated cysts to be used in shrimp hatchery, nursery, and broodstock maturation in China. I am also very proud that the regional Artemia Reference Center is established in China at the Tianjin University of Science and Technology with great support of FAO. The role of ARC is to facilitate sustainable exploration and utilization of Artemia resources in Asia through coordinating interdisciplinary and interregional Artemia research, setting up a platform for Artemia information exchange, providing technical consultation and services for Artemia cyst production and its application in live culture. And I believe ARC will play increasing important roles in the sustainable development of agriculture industry, not only in China, in Asia, but also worldwide. Finally, I wish the Artemia workshop today a great success. Thank you.
Thank you very much, Professor Wang. I see a number of members from Artemia Association of China. So I would also like to take opportunity to extend our appreciation to Artemia Association of China for your great support to the workshop. And now I would like to hand over the moderating task to FAO Rome, Dr. Rodrigo Rubach. Rodrigo.
Thank you.
It's your floor.
Thank you, Professor Lin Sui. Good morning, all. Good afternoon, good evening to all here connected here with us. My name is Rodrigo Rubach. I'm a FAO Senior Aquaculture Officer, Aquaculture Technology and Production Team, and I'm here to act as the workshop moderator alongside with Professor Lin Sui on site. So on behalf of FAO, I would like to thank you, the participants in this workshop, for your presence and time with us here. I would also like to thank all the other organizers for their work. Distinguished participants, a new international interdisciplinary approach is needed to tackle these present Artemia issues and opportunities, like the breakthrough in Artemia use in aquaculture following the 1976 FAO Kyoto Conference. Therefore, the purpose of the GCA Artemia workshop is to explore needs and opportunities for a new international initiative to guarantee a more sustainable provision of Artemia, both from natural sources and from controlled extractive Artemia farming integrated with salt and other fish crustacean aquaculture. We believe in the possibilities of aquaculture, and we trust that your contribution here in this workshop will support the increased economic, environmental, and social sustainability of this important sector. Having said that, I would now like to give the floor to our first presentation by Mr. Patrick Sogelot. Please, Mr. Patrick, the floor is yours.
Thank you, Rodrigo. And let me also express our sincere gratitude to the organizers of the Global Conference on Aquaculture to be able to have the opportunity, in fact, to speak at this Artemia workshop. And let me start to explain what you were just mentioning, what an important role the FAO Kyoto Conference in 1976 has played in the later use of Artemia in fish and crustacean hatcheries, and why, indeed, as you said, it might be time for new actions when it comes to the role of Artemia in aquaculture. I don't think I have to spend much time in introducing this remarkable crustacean, the brine shrimp Artemia, 1 centimeter in size, as you see here, living where the others cannot survive. That means at very high salinity, 3, 4, up to 10 times the concentration of seawater. So the place where fish, shrimp, other crustaceans might not have a chance. To live there, Artemia, of course, has developed some very peculiar characteristics. One of them is the availability, the production of cysts. And in order to explain where these cysts are coming from, let me quickly go through the life cycle of Artemia here. While the baby Artemia, 0.5 millimeter in size, they grow up to 1 centimeter adults under good conditions of temperature, food, etc. And then remarkable, they have two ways to reproduce. They can reproduce by live babies, the live reproduction, as we call it, or oviviparous reproduction. And of course, that means as long as you can close this cycle, that you can start with a small population, grow a big population, that eventually, when the conditions turn unfavorable, might switch to a different way of reproduction. The embryos will start to develop, but at a certain stage, 4,000 cell stage, they stop the development or surrounded by a shell and deposited in the environment as dry cysts. Dry cysts that, as I like to call it, magic powder, that can be stored, can be shipped, but can also be used within a short period of time, less than a day. It can be hatched out into what we call a substitute for plankton, the Artemia nopii, and as we know, suitable food, a good starter food for quite a number of freshwater, marine, brackish water fish, and a lot of different crustacean species. Back in the 1960s, that was in fact the start of commercial availability of Artemia cysts by two companies located in the United States, one in Utah, one in California. And the main market at that time was the aquarium pet market. It was around the same time that the pioneers of modern aquaculture, the Japanese, started to use Artemia in the development of hatchery production of sea bream, kuruma, shrimp, and a number of other species. So 1976, as we just heard from Rodrigo, the first technical conference on aquaculture, well, it was stated there that maybe we have a problem using Artemia in hatchery aquaculture when it comes only from one spot in the world, from the United States, that we need hard currency, in fact, to produce the food, to grow cheap aquaculture products to feed the poor. So we dared at our young age to come up with a number of suggestions for improvements for that, what indeed at that time was a critical Artemia situation. Other sites could be explored. There might be opportunities to introduce Artemia, better techniques, use of Artemia biomass. And it was indeed thanks to the interest and the support of the FAO regional representative Herminio Rabanal that after the Kyoto conference, we got the chance to do a number of study visits and verify if a number of the hypotheses that we had formulated at the FAO conference could be confirmed, could be put in practice. And one of the first was the interest in a region where there was already a lot of good aquaculture and a big interest to further develop hatchery aquaculture, no availability of Artemia. But as some of you know, during certain months of the year, the dry season, there are seasonal salt ponds. And this is where we were verifying that, that possibility with the biological characteristics of Artemia to first, after inoculation, try to stimulate the production of the population, to grow a bigger population, a population that then at a certain moment can be induced to produce cysts. And that's where first in 1977 in the Philippines, later in Thailand, one was able to, through FAO projects, to demonstrate this possibility. It was a very, very interesting coincidence that around the same time, Artemia was introduced in the Nordeste, in an area of a lot of salt fields, where Artemia was not naturally occurring, and that within a number of months, large quantities of Artemia cysts could be harvested. So this was like the stimulus for the Food and Agriculture Organization to believe in the future of Artemia and to recommend the establishment of the Artemia Reference Center, as we did at Ghent University in 1978. We quickly realized that the challenge was enormous because so little was known about so many aspects of Artemia that we launched the international study on Artemia. And you see the spots in the countries where colleagues and other scientists were joining in that ISA. And over the following years and a couple of decades, quite a number, over 50 different papers were published under that common heading, introducing a lot of new aspects in the biology, but also in the application of Artemia. And that was continued in the proceedings of a number of international Artemia conferences, a number of books, and ultimately in an Artemia manual, the first one published in 1986, and then the FAO Live Food Manual with an important chapter on Artemia in 1996. So all at the time when we have seen that hatchery aquaculture was in full expansion, and that in fact, we could say, and in fact, still today, we can say that Artemia is playing a crucial role in the successful aquaculture of a number of these fish and crustacean species. And of course, in parallel, you can expect with the expansion of the hatcheries that the demand for Artemia was increasing at quite a speed when you see the figures here. And good enough. And this resulted as what we see today in quite a number of hatcheries on all continents, small scale, large scale, high industry, responsible for a very important fraction of today's aquaculture. The large quantity of Artemia cysts, lucky enough, could also come from different sources. And for a long time, and still today, the Great Salt Lake, Utah, USA, was in fact, and still is, one of the most important sources, but not the only one. Over the years, quite a number of other sites have been explored, have been developed, and Siberia, Kazakhstan, Uzbekistan, Iran, a number of other countries in Central Asia are very active in Artemia harvesting, processing, and distributing, as well as in China, as we heard from Professor Wang Qingying, coastal areas and inland salt lakes. So this is all the good news, but we also have to be concerned because there are a number of issues at stake. And this is where we should be and are indeed concerned that all the sites and where Artemia is naturally occurring should be harvested a sustainable way. And maybe we need to see in the future how we can come to better agreements and use particular protocols as we will hear in a moment from Thomas Bosteels on all the efforts that were spent in the past decades on ensuring a sustainable harvesting at the Great Salt Lake. We need also to protect these Artemia habitats because there are already indications we will also hear about later, climate change impacts, number of salt lakes that are disappearing. And on top of that, assuring that we preserve the Artemia species gene pool, the number of species, the number of strains that are existing. Coming back to that original work and the early pictures that you see here from the first successful inoculations, it's good to see that over the years, a quite standardized protocol, we will also hear about later in the presentations, but in a number of tropical climates, be it in Asia, but also, on other continents, during the dry season, the salt production can be complemented with Artemia. And there you have the Artemia cysts and the Artemia biomass. A very interesting new development that started first in Thailand, later in Vietnam and a number of other countries. Brine waters, 3-4 times the concentration of seawater, or an ideal water to dilute with fresh water and make up the convenient brackish water, seawater for use in inland hatcheries that might be remote away from the coast. That's in the salt in the dry season, Well, in the rainy season, since there has been a conversion of the ponds to allow that integration with Artemia. During the rainy season, there is an opportunity to develop other species. So we have seen, and this is something we are very hopeful that in the future, local availability of Artemia can be more stimulated, can be more considered, and in fact, could then be the catalyzer for a number of new aquaculture developments on different continents, contributing to more seafood security and, as we will also hear later on, providing better socioeconomic conditions, particularly in regions where the artisanal salt making is now under very much stress, and in fact, in many places even has resulted in subsistence survival. Another thing that maybe is just typical for Vietnam, where the salt farmers got the idea, Artemia biomass, is that not something that we could consume? So the Artemia omelette, a popular product in the salt field areas in Vietnam, but why not consider this as a potential ingredient, high nutritious ingredient in human diets? Low in the food chain, Artemia is feeding on microbial biomass, on bacteria, on microalgae. We couldn't see a more sustainable opportunity, a more sustainable source of animal protein production in the aquatic environment. So at one of the last opportunities we had, November 2019, as you see there, a number of us, all panelists and speakers here in this workshop, were sitting together in Kuala Lumpur thinking about, is it not the time to look into some new initiative? Shouldn't we give a revival to some of the activities in Artemia? And that's where the idea of the International Artemia Aquaculture Consortium was born, and where I must say also thanks to COVID, we were forced, in fact, to look into other opportunities to interact with people. And through meetings, email, video conferences, I'm very proud to say that we have already an engagement of 43 partners from public and the private sector, many of them probably attending also this workshop today from 28 different countries. We will hear in a moment about the outcome of a first international workshop that was organized, focusing on Artemia pond and tank production. We were wondering, as members of this new international community, well, hatching out Artemia, that's not a difficult thing, but all the parameters, all the conditions that have been described and that were worked out in manuals and in papers, are they well followed up? Or are we still applying the good aquaculture practices? Do we know about the different steps in the hatching process, the risks that might be involved, risks from a nutritional point of view, risks from a biosecurity point of view, the different forms under which we heard it already from the introductions, that under different forms Artemia could be used. So this is where just a few weeks ago we had a very interesting webinar organized by NACA, and where you will also hear the outcome later in this presentation. No time to go into more details, but I just like to bring to your attention that also over the past decades, Artemia has become a very unique model organism and is in fact used in a lot of research, as you see listed there. And let me just draw your attention to maybe the most spectacular one, the last one, the cell cycle studies, where a colleague from China has been able to identify the gene in Artemia that is responsible for the stop of metabolic activity, to go in that arrest of metabolic development, and that with that gene, he can indeed do transplantations in cancer cell lines and switch on and off the cancer development. Or in other words, you see how our brine shrimp Artemia can be also the subject and the model organism in quite a number of studies. And I think we can expect, in fact, a major progress and development in not only that research, but also for applications in aquaculture, when we will hear in a moment about the availability of the annotated Artemia genome. So that brings me to my final slides on what could be the activities that we could consider for that new consortium, and that, of course, should be seen now as a first suggestion for further discussion. And I repeat a number of the topics that I brought up already. Conservation of Artemia biodiversity, very important that we learn more. We have a lot of knowledge, but we need to learn more about Artemia biodiversity and look into what probably will also be explained in more detail. We need to consider now wild and farmed species of Artemia. Sustainable harvesting of the wild resources I was referring to, very important that we see what type of protocols, what type of research is needed to bring this to a good result. Integration of Artemia and aquaculture production in artisanal salt farms, very clear and evident. With all the news we hear about climate change and climate impacts, what could it have as an impact on Artemia in inland lakes and in coastal saltworks? Strain selection and selective breeding, things we have had very, very little experience with in the past, but opening now also quite a number of new opportunities. If we think, for example, about using Artemia as a protein source, an animal protein source, maybe we can select for bigger Artemia strains, where in the past the main interest was to go for smaller cysts used in aquaculture. You will hear that it's really timely that we consider to go back to good aquaculture practices in the use of Artemia in the hatcheries, that we might consider also that form of extractive Artemia, low in the food chain, maybe to integrate it with other aquaculture activities. And as I mentioned earlier, the use of Artemia biomass maybe in human diets. So 45 years after the Kyoto conference, I think this is the appropriate time to launch this new initiative. And maybe it is also the time to pass the torch to the next generation of Artemia specialists. Thank you.
Thank you, Patrick. That was very insightful presentation. I really appreciate all these topics covering from Artemia and really align with the SDGs and how we can move forward within this consortium. So thank you for that almost a small lecture here on where we are at and where we should go towards more productive Artemia in aquaculture and even for human consumption. So that's very enlightening. As for the next presentation, I'd like to call Mr. Mezanur Harhar from World Fish Center in Bangladesh. Ms. Mezanur, the floor is yours. Thank you.
Good evening. It is nice and a very pleasure to have a presentation in such a big international workshop. My name is Mizanur Rahman. I am from Bangladesh. Next slide, please. Yeah. International workshop on Artemia on pond or tank production organized by World Fish in the framework of European Union DCIRA project named Artemia for Bangladesh. The conference was contributed from different countries in Asia and Africa. More than 70 participants from 14 countries participated. Next slide, please. The major aims of the EU project are to introduce integrated salt and Artemia production in the dry season and new aquaculture activities in the rainy season. The purpose of the workshop was to exchange experiences in Artemia pond and tank culture among interested stakeholders and stimulate information among the participants. Next slide, please. As you heard in the opening talk of this workshop, already in 1976 at the first FAO Aquaculture Conference, the possibility was raised to consider introduction of Artemia in suitable locations, for example, in seasonal salt works. Indeed, in many regions in the world, seasonal salt production is practiced, and one could combine the production of salt with brine shrimp Artemia. Once pond salinities are sufficiently high to keep Artemia predators out, Artemia nuclei can be inoculated, and within few weeks' time, adult biomass and cyst can be harvested. Next slide. The country, Vietnam, is very successful in developing models for Artemia cyst and biomass production in ponds. The photographs showing activities of Artemia cyst and biomass production. Right now, Vietnam is producing 20 to 30 metric tons cyst per year in about 1,000 hectare area. Next slide. It helps salt farmers' production income increase 3 to 5 times compared to salt production due to Artemia culture. It can be further increased if intensive models are used. Next slide. So, small-scale farmers in Binh Chao and Bac Lieu provinces in Vietnam, and 2 companies named Binga and Kim Juan involved with Artemia culture. The annual revenue reached $4 million US. Thank you. Next slide. In India, Artemia are supplementary fed with microalgae. You can see the left side, fermented rice bran and microalgae, and they are also successful in producing decapsulated cyst, Artemia cyst, flax, frozen biomass. And the culture area is in Tamil Nadu in India. Next slide, please. In Iran, the present production reached to 8 metric ton cyst and 30 metric ton of biomass. The Artemia species are Artemia franciscana, Artemia urmiana, Artemia parthenogenetica are used for cultures. And in tank production also started in Iran. Next slide. In Kenya, as you can see that also in Artemia production has been started. The present production reached to 38 kg cyst and 140 kg for biomass. The photographs showing training and demonstration activities in Kenya. Next slide. In 2016, Artemia culture started in Cambodia, and production reached 3 kilo cyst and 84 kilo biomass in 2017. Next slide. In Myanmar, you can see the photographs of Artemia cyst hatching, Artemia biomass harvesting, Artemia cyst processing, Artemia pond racking. Myanmar also started Artemia inoculation first in 2014. Next slide. In Bangladesh, you can see that in-house training stakeholders, Artemia training together with international consultants from Patrick and Professor Hua from Vietnam, consultation meeting in the field, first successful Artemia farmer in Bangladesh, Artemia nuclei inoculation at Eden Star 1, Artemia biomass harvesting and biomass processing, and even Artemia cyst processed also locally concentrated brine. Next slide. In China, 4 species are cultured: Artemia franciscana, Artemia sinica, Artemia tibetania, Artemia parthenogenetica. China has been producing huge amount of biomass, 60 to 70,000 metric ton of biomass, and mainly produced semi-intensive pond culture. Artemia biomass are used for aquarium fish, shrimp nursery, broodstock maturation, feed ingredients, and shrimp farms. Next slide. In Thailand, Artemia mainly produced Thailand produced mainly Artemia biomass. You can see that biomass processing, biomass preservation in Thailand. Thailand biomass production reached to 340 metric tons. Next slide. In Malaysia, palm kernel expeller, a cheap agricultural byproduct, are used for Artemia biomass feed, and biomass are mainly fed to ornamental fish in Malaysia. Next slide. So the opportunities and challenges for Artemia culture are: integrate Artemia fawn production in artisanal salt farm on different continents. Consider Artemia pond production in desert or arid areas or on salt-affected soils. More work on species or strain selection, improvement for aquaculture applications. Make Artemia pond culture more resilient for climate change events. And select suitable agricultural byproducts as food source for Artemia. All these informations are available in the website of International Artemia Consortium. Thank you all.
Thank you, Mr. Mezanur. That was very enlightening information that you presented, and nice to see the development that has been going around Southeast Asia and Asia in general. And I think that's pretty much complements the previous talk from Professor Patrick. So thank you again for that presentation of yours. I also like to point out that if the audience has some questions, we might be able to answer some of those during the part in the question and answer by the end of the presentations. So with that, I would like to give the floor to Simon Wilson, which will present a report from the status of the use of Artemia cysts in fish and crustacean hatcheries around the world. Thank you. The floor is yours, Simon. Thank you.
Thank you, Rodrigo. So I'd like to present a report of the NACA webinar status on the use of Artemia cysts in fish and crustacean hatcheries around the world, which was held on the 2nd of September. The main aim of this webinar was to review the differences in practices with Artemia cyst hatching and use as food in commercial hatcheries. The webinar considered both large and small hatcheries from different countries in Asia, Europe and Latin America, which were producing a variety of species including pinnate shrimp, macrobrachium prawn, mud crab, European sea bass and sea bream. I will not be able to go into much detail, but please refer to the NACA YouTube channel where you can find the recordings of all the presentations. So we were interested to see if the techniques that were recommended in the Artemia handbooks and manuals published in the 1980s and 1990s are still practiced to ensure the optimal use of this costly resource, particularly at a time when there are many different Artemia sources in the market from the historically important Great Salt Lake in Utah, but also more recently from many other Artemia sites in Central Asia and coastal China. Hatching Artemia cysts is a simple procedure. After 24 hours of incubation in seawater, the nauplii hatch from the cysts, but when conducted in kilogram quantities in commercial hatcheries, there are many aspects in this hatching process that should be optimized to give the best results. Let's have a look at some of the many examples that were presented in the workshop. Culture systems around the world range from small conical tanks of a few hundred litres up to the large concrete tanks as seen here in Chinese hatcheries of 5 to 10 cubic metres in volume. We notice different conditions of illumination of the water surface in the tanks, which is a very important parameter to ensure maximum and synchronous hatching. Many facilities operate without temperature control, resulting in fluctuations of several degrees Celsius between the seasons, which has an impact on the instar stage at harvest, as most of the time the same 24-hour incubation period is practiced throughout the year. This means that during the hottest season of the year, one is mainly harvesting instar 2 nauplii that are contaminated with Vibrio bacteria. This represents a loss of 10 to 20% in nutritional and commercial value over instar 1 and introduces significant biosecurity risks. Here are two examples of top-class Artemia rooms. To the left is a marine fish hatchery in the Mediterranean, and to the right a shrimp hatchery in Vietnam where conditions are strictly controlled and where large quantities of cysts are hatched out on a daily basis. Today, some of the largest shrimp hatcheries in the world process up to 100 kilograms of Artemia cysts on a daily basis. Prior to hatching and incubation, different techniques are practiced for cyst disinfection, for example with hypochlorite or hydrogen peroxide. Some hatcheries are still applying the cyst decapsulation technique with hypochlorite at high pH. While this does indeed disinfect the cysts and facilitates the later harvesting of the nuclei, it is not an environmentally friendly or healthy process. In the top row, we can see a few more Artemia rooms. Underneath are examples of common methods used for harvesting the nuclei and separating these from the remaining cysts. Some apply double screen separation, and very often the nets are not submerged during this harvesting and washing process. These methods cause physical damage to the nuclei, resulting in losses in nutritional value, and the leached contents are a suitable substrate for Vibrio bacteria development. As remarked earlier, this results in further losses in quality and increased biosecurity risks. Separating the last empty cysts and the shells from the free-swimming nuclei is not easy. That's why we hear about innovations in Latin America as well as in China with new separation techniques using brine or hydrogen peroxide. Heat killing or freezing the freshly hatched nuclei in order to be able to feed the shrimp at an earlier larval stage is a valid option, provided that the quick freezing is performed in thin layers, or short heat killing to prevent the rupturing of the thin exoskeleton of the nuclei and the leaching of their body fluids. Again, the same warning as we made earlier, and another example of how one can waste this valuable live food source. The practice of submerging the Artemia in boiling water, as seen on the left, or freezing in large bags is not recommended. More recently, a very elegant separation method has been developed that takes advantage of the possibility to use magnets to separate unhatched cysts and shells that have been coated with iron during the cyst processing. Small hatcheries are using these popular blue tube separators filled with magnets, and more recently magnetic bar systems have been developed, allowing further automation and intensification of the nuclei harvesting process. Here is a model of an Artemia hatching room that ensures maximum output of the highest quality nuclei, processing 100 kilograms of Artemia cysts on a daily basis in 3 shifts. As you can see in the black and red time bar, by improving its former protocol and tank setup, this shrimp hatchery in Vietnam can conduct Artemia hatching under optimal conditions for light, pH and temperature. This has resulted in a 30% more efficient use of Artemia and harvesting a more biosecure product, ensuring a maximum percentage of Instar-1 nuclei. In between feeding times, the nuclei are stored at 4 degrees Celsius to slow development. Marine fish hatcheries in Europe were the first to adopt cold storage of Artemia nuclei and now use milk storage tanks, as you can see in the picture. The technique of cold storage of Instar-1 nuclei needs to receive much more attention as it allows nuclei to be maintained in their most nutritious condition following the morning harvest for the rest of the day. It also allows more frequent distribution to the fish or shrimp tanks, including through automatic pumping devices. Vietnamese crab hatcheries made a big breakthrough when they found out that they could replace the Rotifer brachionis, a classic starter food for the zoea stages, with the umbrella stage of the small Artemia cysts produced in the Vinh Chau salt ponds in the Mekong Delta. Thanks to this innovation, several hundred crab hatcheries in the Mekong Delta now produce more than 1 billion crablets. It is interesting to mention here that the first use of umbrella Artemia in zoa shrimp feeding has also been reported from Vietnamese hatcheries. The production and separation of the umbrella stages during the Artemia cyst hatching process is still a primitive and labour-intensive method, but for sure we can expect to see some innovations here. Over the last years, companies have begun to specialise in the production and distribution of live nuclei, particularly to shrimp hatcheries. The success has had a lot to do with the earlier cited problems of unreliable quality of Artemia norplii produced under suboptimal production conditions in so many hatcheries. In China, the biggest consumer of Artemia cis, close to 50% of the shrimp hatcheries rely on the purchase of live Artemia from these norplii production centres. Finally, we need to mention the growing use of Artemia biomass harvested from production ponds for use in late hatchery and nursery feeding, especially in shrimp farming. China is a leader in this practice where biomass is offered in live, frozen and dried forms. So this brings us to the conclusions of the NACA Artemia webinar, organised on the 2nd of September. In the first place, it's very clear that there is a lot of room for improvement and that it is high time to reconsider good aquaculture practices for Artemia production and their use in the hatcheries. Hopefully, hatchery managers will realise that applying more standardised protocols will not only result in a better and more biosecure food, but that they will be able to save on their Artemia cyst purchases. All efforts need to be made to optimally use Artemia to guarantee that hatcheries deliver top quality product for stocking in the ponds or cages. FAO has therefore decided to prepare an update of the Artemia manual. We also recommend to consider holding regional Artemia training courses for local hatcheries. The use of umbrella Artemia as successfully applied in the Vietnamese crab hatcheries is an interesting new development that should be considered for wider application in aquaculture as new source of food in the early larval stages, be it for shrimp or fish. In view of the large variety of species and strains of Artemia that are now available in the market, it might be time to study their specific characteristics so as to identify their most suitable application for specific species of fish and crustaceans. Such could relate to their nutritional composition, synchrony in hatching, or enrichment characteristics. Finally, it might also be worthwhile to reconsider a wider use in hatcheries of the Artemia enrichment technique, as it is now restricted to applications in marine fish and crab production. This method not only allows enhancement of the nutritional value of the norplii, but can also be used as a vector to deliver, for example, pre- or probiotics to the larvae. Okay, and that's it. And thank you for your attention.
Thank you, Simon. Very interesting presentation as well, and really aligned with all the work necessary, and also lots of technicalities and technical expertise reported here. So we sure need to put that together in a way to provide better training and better advancements on this specifics for the Artemia culture. Thank you much. So with that, I'd like to ask for the presentation from Mr. Thomas Bostius from Great Salt Lake Brine Shrimp Cooperative, USA. If you please, Mr. Thomas, the floor is yours. Thank you.
Thank you very much, distinguished guests and friends and colleagues. I do thank you for the opportunity to present how we manage a hypersaline resource here in Utah, namely the Great Salt Lake. Next slide, please. Just by way of introduction, what I'd like to do is just briefly discuss the worldwide Artemia resources. We do believe, of course, that man-made resources are going to become more and more important in order to sustain the growth in marine aquaculture. That being said, with at least more than 90% of the commercial Artemia today still coming from natural resources, it remains really important that we manage those natural resources in a sustainable manner. Next slide. So we know that there are many anthropogenic influences on our resources. Some examples are the unregulated harvest, salinity increases, nutrient losses, and water shortages. Close to our home here in the USA, we have the example of Owens Lake, where water diversions for Los Angeles in 1913 already resulted in a dry lake bed by 1926. And we know that we see many of these issues worldwide, and frankly, we see them also on the Great Salt Lake here in Utah. Next slide, please. So the question begs how we tackle these multiple issues related to our endorheic lakes. And what I would like to do is give the example of how we've been working on these issues here on the Great Salt Lake. By no means do I mean to infer that that example should be used in other natural resources. I do believe that every hypersaline system is unique. It has a unique set of circumstances, and it often needs a unique approach to sustainable management. But maybe the Great Salt Lake can serve maybe as an example, if you will, of how some of these issues could be tackled. Let me start with talking about sustainable harvest management here on Great Salt Lake. In the early 90s, there was truly an increased harvest pressure on the Great Salt Lake. There was really no formal program to manage the resource. There was not a lot of research being done with regard to the Artemia resource, and the industry was frankly becoming very concerned about overfishing of the resource. Next slide. So the industry then took the initiative, approached the Utah Division of Wildlife Resources with those concerns, and the division created the Great Salt Lake Ecosystem Project. It's a project that's tasked with managing the resource and studying the resource. The division also created a technical advisory group. It's a peer review committee that helps guide that research. And then the division actually increased the industry fees to harvest that resource, but then used those fees to work jointly with other research institutes to improve the research of the resource. Next slide. With that data, the state, and with the support of our industry, the state then created a sustainable management model. It's a model that's based on a modified stock recruitment curve, a modified Ricker curve, if you will, where we try to leave an optimal amount of cysts in the lake after the harvest in order to assure an optimal population growth in a subsequent season. That model takes into account many different factors, and it ultimately created a very sustainable management model that allows the industry to actually harvest the excess cysts above 21 cysts per liter. Next slide. As you can see in this graph, since we've implemented this model, what we've noticed is a decrease in the variability of the harvest and an increase in the actual dry equivalent harvest. And that has been, for us, an indication of the success of the management system. Next slide. So with management under control, it brings us to the questions of salinity and nutrient management. And salinity and nutrients are two main drivers for the Artemia population health here on Great Salt Lake. With a couple of decades now of research, what we can say is that the optimal salinity for the Artemia population on Great Salt Lake is between 120 and 160 ppt. That the lake is primarily nitrogen limited, although there is a co-limitation of nitrogen and phosphorus, and that the Great Salt Lake really has a significant excess capacity for assimilation of nutrients. Next slide. So with that research in mind, what we then know is that if we can keep the salinity within a safe range, higher than 120 ppt to avoid predation and problems with phytoplankton communities, lower than 160, 180 ppt, where we see significant population level declines because of physiological reasons primarily. If we can keep that salinity within that optimal range and we can have adequate nutrient loading into the lake, then we know today that we can keep that Artemia population at present levels, even with a highly reduced volume of Great Salt Lake. Next slide. So the question then begs, how do we manage that salinity? Well, on the Great Salt Lake, we're fortunate that we have a causeway that bisects the lake, that the major drainages into the lake are coming into the southern part of the lake, as shown on this map. And that creates a strong salinity gradient between a more saturated north arm and a less saline south arm on Great Salt Lake. There is a breach in that causeway that allows for bidirectional flow between the north and the south. And there is now an adaptive management berm in that breach that actually allows us to regulate that bidirectional flow and therefore also regulate the salinity and manage the salinity of the south arm of the lake, which is the primary Artemia resource. Next slide. So with the ability to manage salinity, then the question becomes, what are we doing about nutrient inflows and about nutrient modeling on Great Salt Lake? In Utah, it is the Division of Water Quality that is developing nutrient criteria to protect the beneficial uses of Utah waters, and that includes the Great Salt Lake. That division actually created an advisory team, and a technical team that guides that process and takes into account the needs of many of the stakeholders. And the Great Salt Lake Ecosystem Project is also working and studying nutrient inflows and more recently also nutrient cycling. And so we're seeing a lot of progress that's being made today in both the management of these nutrient inflows into the lake, but also the modeling of the nutrient dynamics in Great Salt Lake. And what we know today is that at least 90% of nutrients within the Great Salt Lake ecosystem are cycled between the different trophic levels of the lake. Next slide. So with salinity managed, nutrients under control, the question then begs, what are we doing about lake levels? And we know we are seeing problems with our lake levels today. The lake levels are decreasing. What we have learned on Great Salt Lake is that that decrease is, the reason for that decrease is the consumptive use of water. It's the use of water for agriculture and for development. And that has lowered lake levels by 3.6 meters over the last 150 to 200 years or so. And in fact, if it were not for that development, then the lake today would be at the same level, exactly the same level as it was in 1856, as shown on this graph with the blue line. Next slide. So we do have issues with water inflows, and the question then becomes, how are we dealing with that? Well, in 2010, the Utah legislature actually enacted by law the Great Salt Lake Advisory Council. It's a council that advises our governor and also advises several of our departments in Utah. And it advises those on sustainable use and development of Great Salt Lake. And today the council has focused a lot of efforts on water management, and that has already resulted in changes in water laws that actually allow to maintain water inflow into the lake, federal infrastructure investments to help preserve the Great Salt Lake ecosystem. And last but really not least, also, it has truly increased the public awareness about the importance of Great Salt Lake to Utah. Next slide. So what did it take to create this sustainable management of Great Salt Lake? Well, it took a real cooperative process with many of the stakeholders. It did start with our brine shrimp industry, working with the Division of Wildlife Resources, but that division is now studying many different aspects of the lake. It took the formation of the Great Salt Lake Advisory Council that's now really focused on water conservation and water strategy development. The work of the Division of Water Quality, focusing on standards for nutrients, the work of the Forestry, Fire, and State Lands Department that is focused on the management of salinity. And really, today, it's not just a brine shrimp industry, but many other stakeholders, industries, and organizations that are participating in this process. And really, I wouldn't do it justice if I didn't mention also all of the universities and research institutes that are supporting the research here on Great Salt Lake. Next slide, please. So then the question is, did we succeed in creating this sustainable management system? Well, the truth is that this is a very complex and a very dynamic system. there will for sure always be more work to be done. But with 2 decades now of scientific research and cooperative management, we can say that we have implemented a sustainable harvest management. We now have a tool to manage that salinity. We're confident that our nutrient inflows can be managed, and with salinity and nutrients being managed, we know that our Artemia population is shielded from significant water volume declines, contaminants are under control, and there is now a real focus on managing water inflows into the lake. And with that, I thank you very much for listening.
Thank you, Thomas. Very nice presentation, really informative. And thank you for sharing that experience. And very important to give some lights on further how to go with those ecosystems and how possible solutions in that side as well. Thank you very much for that. And with that, I'd like to call for our next presentation, which will be from Mr. Gonzalo Garrado from Los Lagos University, Chile. Mr. Gonzalo, you have the floor, please.
Thank you very much. Good evening in China. I'm going to present a quick account of Artemia genetic resources in the world. Next, please. What I would like to highlight here is that we have 6 regional sexual species and also a lot of asexual types distributed in the world. But what is most important is that most of these genetic resources are harbored in locally adapted populations. And Artemia franciscana is a very well example of that. I will talk about that later. And also we have incipient farm types, as it was mentioned before. Next, please. Well, what are the challenges? One is lack of knowledge to assess climatic change and other perturbations. There is another problem that the genetic variability is losing at alignment rates. And you know that genetic variability is really important for species or population resilience. So this is a worrying trend. There is a need of systematic monitoring of genetic diversity. This is to comply with the international guidelines, FAO, for example, and CBD, that means Convention of Biological Diversity. And I think, and this is to be discussed, there is no policy or institutional framework for monitoring and protecting wild intraspecific genetic variability. Next. Okay, these are the 6 species. You know them very well. Artemia persimilis is the oldest, and we are lucky to have it in Chile and Argentina. What is important here is that each species is a unique combination of genes and interaction with the environment. Each has a unique evolutionary story. So I think that one of the main problems is that Franciscana is the best studied one, but we need to study all the other species very well because we are to find novelties in each one. On the right side, you see the tree that only shows that using different new nuclear or mitochondrial markers, okay, you get always the 6 species. There are some populations that normally do not match with the species, but this is something that we could discuss in another opportunity. So what is important is that all the populations are locally adapted. This is the example of Artemia franciscana. the most widely distributed species. You see here on the right, there is a wide latitudinal variation. All populations are different genetically and phenotypically. And that means that maybe they have aquaculture traits or adaptive traits that are unique, but we need to discover them. The other point that was already mentioned is translocation to different places in Macau, for example, Brazil, Vietnam, China, Thailand, I saw Kenya. This is very important because we have the origin of farm types. This is something that we need to continue monitoring. What is the difference between a farm type and a local wild population? And what is really important to understand what means locally adapted population are the studies on genomics and transcriptomic studies that we hope will provide clues on adaptive and productive genes. Okay, this is the Vinh Chau farm type, Vietnam, that came across after San Francisco Bay cysts were introduced in Vietnam. In 1996, I saw from Professor Sorghilu's presentation that cysts were introduced in Macau, in Brazil, earlier on. So we don't have much information about the quality of those cysts. What is important here is that selective pressures in Vietnam selected individuals with tolerance to high temperature. And what is known so far is that heat shock proteins are upregulated, are differentially expressed. But as you will see later on, there are so many genes that can be upregulated depending on the environmental conditions. Next, please. Yes, thank you. So here you have the environmental component of the reproductive mode. I consider this because of the importance for survival of the species and populations, but also because it has a lot of productive importance. On the genetics side, well, you will see later from Gilbert, Artemia genome has a lot of genes, as many as humans. So I was impressed to see that. So you have a very complex, you see in the bottom of the figure, genetic environmental interactions. And this is really a black box that we need to understand. And I think Artemia is a very good material to do this, even under laboratory conditions. On the left side, well, the genetics of the mother could be important in perceiving the environmental change. We, in very early stages of the genetic knowledge of Artemia in 1998, showed that females with multiple genes with high variability or heterozygosity were able to produce more cysts. So you see here the genetic makeup influences the ability to produce cysts. So I see with good eyes the managing work that Thomas is doing in Great Salt Lake to maintain the ability of females to produce cysts. But what you see in the third line on the left, there are more than 300 genes controlling cyst diapose and post-diapose functions. That's a lot. If you see the interaction of each gene with a particular environmental condition, you will be amazed the difference that can appear. For high salinity tolerance, there are more or less 600 genes, and this is also impressive. And for anoxia tolerance, about 900 genes. So if you think in the interaction of these genes with the environment, okay, we have a great difficulty. So this slide is mainly to convince you that we need to improve the characterization of different wild populations of Artemia and also of farm types. For example, what is the difference between the Vietnam strain and the Macau strain? Next, please, Rodrigo. Okay, what are the problems? Well, we have a mandate to protect Artemia natural habitat and their biodiversity, because these resources are source of novelties for aquaculture. But what is happening? The saline lakes are declining around the world. As you see here, they are shrinking at alarming rates. And Thomas mentioned about Great Salt Lake. The photo on the left is Great Salt Lake, published by this author. And then you have Urmia Lake. I think, Patrick, that we did, we took that boat when we were in Urmia. So you see the boat there. And also it's very interesting to see what is happening in Ariel Sea. Thomas talked about that. So next. But as important as the loss of species is the loss of genetic variability. So I put here a graph that I think is very important because it illustrates us the type of information that we need to have. Here you have a contemporary heterozygosity on the left, and in the bottom you have historical heterozygosity. That means that they collected the ancient DNA. And over the line, you have the genetic diversity gains. And below the line, you have genetic diversity losses. So what is important, if you see all these species that are here on the top, with different kind of classification according to IUCN, the conclusion is that they are losing genetic variability. And if they are doing so, they are losing their ability to persist in the environment. Next, please. One of the studies I know, besides Great Salt Lake, that is being monitored for more than two decades, is Artemia urmiana in Urmia Lake. So after 10 years, next please. Oh, sorry, it didn't appear, but author showed here that polymorphic loci are losing variability after 80% of reduction of the surface of the lake. So finally, next please. I think it's important what I said regarding establishing, strengthening national global characterization, monitor, and information system for Artemia genetic resources. This is a mandate of FAO for all species, and we really hope that you realize that we have very few information about all the populations composing the different Artemia species. It's important to have information for detecting diversity losses over time. And here is very important to have cyst collections from around the world. So it's very important what Li-Yin Su is doing with the Artemia reference in Asia, and also what the Artemia Reference Center in Belgium has done in the past. We need to assess differences between farm types and local populations, and we need to coordinate actions to comply with international guidelines. I think the Artemia genome that is recently published will facilitate the study of interspecific genetic variation, identification of key adaptive genes. I mean, there is a very good opportunity to do genotype environment interactions, and Artemia is a very good model to do that sort of studies in the laboratory, and also could be done in the wild. Okay, thank you.
Thank you, Mr. Gonzalo Delgado. Very informative topic. Thank you for reminding us that how this importance of these studies in genetic diversity and how we can fulfill that mandate for us, especially for the SDGs and the going in the same direction as the CBD, the conventional biological diversity of those important natural assets that we have. Thank you for that. And with that, I'd like to call for our next presentation from our colleague Gilbert van Stempen from Ghent University, Belgium. Gilbert, you have the floor, please.
Okay, thank you, Mr. Chairman. Good evening to the people in the room in China, especially. And in fact, Gonzalo's presentation was already a good transition to my presentation, because he already mentioned about the Artemia genome. So this long-awaited for publication has been published recently. You find the title page there. It has been published in the journal. At the left-hand side of the slide, you can see it, BMC Genomics. It's open access. At the right-hand side, you see that it has many authors from different affiliations, too many to mention here, but I just want to give credit to the name, which is in the blue circle, Stephanie de Vos, the first author. She really did an enormous job with a high dedication for years to bring this undertaking to a good end. So next slide, please. So the paper is about the annotated Artemia genome. What is a genome? We know all this. The genome is actually all the DNA of an organism, all the genetic information. There have been efforts in the past, published in literature, to describe the genome, parts of the genome, the transcriptome. But okay, this is the most comprehensive, the most complete effort so far in describing the Artemia genome. It's not 100% complete yet, but it's rather complete. So the next slide, indeed, what is genome annotation then? Well, as the first line says, annotation is a process of identifying any functional elements of the DNA sequence. With other words, identifying genes and what these genes are doing. And just to highlight the significance of all this, I borrowed two definitions from literature that I found, saying first, the annotation actually gives meaning to the genome. And the second quote there, I even like it more, annotation bridges the gap from, let's say, the naked sequence of the DNA to the biology of the organism. Annotation tells us what the organism is and does and how it functions. So next slide, please. Of course, yeah, no time here to go into detail, but from left to right, DNA has to be sequenced, has to be read. For this purpose, it has to be cut into pieces, shearing this is called. Then these small pieces, relatively small pieces, are sequenced, they are read, and then the whole original text, so to speak, has to be assembled again, has to be reconstructed to its original sequence. That's the assembly. Needless to say, this comes with quite sophisticated bioinformatic tools. You need quite some skills to bring this to a good end. And then next comes the annotation. And this is then identifying with which genes the sequences correspond and what these genes are doing. Again, this comes with it bioinformatic tools, comparing with databases of other genomes, etc., etc. And well, Gonzalo already brought this up. Through this exercise, indeed, almost 22,000 genes, 22,000 genes could be identified. And this corresponds with a more or less 90% assembly of the genome. So if we have the entire text, so to speak, 100% of the genome, 90% has been reconstructed. So as I said a while ago, the exercise is not complete yet. It still needs fine-tuning and completion. So next slide, please. So we can use this database now to describe processes and pathways, just as people have been doing in the past for plant models. Arabidopsis used to be a plant model to describe them, to study the genome of crop plants, such like maize. So in the same way, we can use the annotated Artemia genome as a model for crustaceans, including aquaculture crustaceans. We know that the genome of white shrimp, Lithopenes fanamei, is available as well. We also know that Artemia is a so-called extremophile organism, so living in a very specific environment with all kinds of interesting features. So the paper is also describing the Artemia genome with, for example, that one of white shrimp, with the genome of other invertebrate organisms, and the paper is describing what the Artemia genome has in common with those other genomes, and also with what is specific for Artemia. This means as well that a variety of processes, traits of interest, you find them there at the bottom of the slide. The list is not complete. They can be studied through what is called gene discovery, as the title of the slide says. So trying to identify genes that correspond with certain characteristics of interest, that get into action, that are expressed in certain conditions. And the next slide, please, gives one example of this. There are several examples of this in the paper, very interesting examples. But for instance, the authors are analyzing which genes are expressed in two different salt conditions. Standard salt condition, 30 grams per liter, and high salt conditions. And then, well, as Gonzalo already mentioned, the authors found that 670 genes were differentially expressed. And by the way, of these 670, 32% have no known function yet. We just see that they are differently expressed in the two conditions, but a whole lot of them, we don't know actually what they do. So there is still work to do here. So I'm coming to the end of my slides. So where is all this information available? It has been uploaded on an interactive online platform, Orcai. It has nothing to do with a whale species, but okay, it's a letter word for online resource, etc., etc. It is operated by, summarizing in the blue circle, the Flemish Institute of Biotechnology that is linked to our university. This public, on this database, several public genomes are available. You see a few there, they are arranged alphabetically, but ours, Artemia, next slide, please, is also uploaded there, and you can browse it freely. It's open access, so to speak, just to read, to consult it. However, if you want to change, you can work in it. It comes with a manual, of course, quite an extensive manual. And if you want to work in it, you need to have an account. This account can be asked for by simply contacting the head of our laboratory, Professor Peter Bossier, just explaining the work that you intend to do. And of course, we are sure that, as previous speakers have been saying, that this Artemia genome is indeed a very powerful tool to make progress in a number of the activities that have been listed by Patrick Sorgelos and also that were brought up by Gonzalo Carrera. Thank you very much.
Thank you, Gilbert. Again, a very interesting presentation and give peculiar viewpoints to pursue and be aware of the potentialities for these interesting species as throughout the knowledge base that we have and still don't know much. So with that, I'd like to bring to our, since we are, I think we are still on a good time, we are still on schedule, so I'll bring the questions and answers for this session now. And with that, I would like to ask our Professor Ling Sui, as my counterpart in presence there in Shanghai, to seek some questions from the audience. Then I move on for other questions that we might have from the question and answer box here. Professor Ling Sui?
Yes.
If you please.
Is there any questions from the audience here? Also.
I'm Gao Song from Artemia Association of China. Thank you very much to all specialists. You had given us a so interesting presentation tonight. I have two questions. The first question may to our good friend Thomas in the United States. So biosecurity is a very important issue in the hatcheries that we are using live food Artemia. Except for controlling the hatching of seeds and harvesting condition of monopoly in the hatcheries, are there any effective measures need to be conducted during dry seeds processing to reduce the pathogen contamination? That's my first question. I have the second. My next question is maybe to Professor Gonzalo. Yeah. My second question is, we have a lot of parthenogenetic Artemia in China. Parthenogenetic, yeah, Artemia in China. Do you expect that these are all different strains with different characteristics, or how similar may they be, especially when it comes to their application in aquaculture? Thank you. That's all my questions.
I'll ask Gilbert to go ahead and, forgive me, for Thomas to go ahead and answer the first question, please.
Sure. Yes, Gao Sung, very nice to see you again after all those years.
Yes.
Yeah, I'll try to answer that question as best as possible. What I can say is that I believe biosecurity actually starts with the actual resource. So, for example, I'll take the example of the Great Salt Lake in Utah. In Utah, around the Great Salt Lake, we actually do not have any marine fish culture, any shrimp culture. So we are already isolating and creating a biosecure environment, if you will. And I think that's where it starts. Obviously, the high salinities of our hypersaline systems help us with controlling some of the pathogens that we typically see in aquaculture. In terms of the processing itself, I believe that with the right disinfection during processing and with the right protocols in place, at least what we have seen is that we can actually process a cyst that does not contain either human pathogens or, for that matter, aquaculture pathogens. Specifically to our organization, in the last 16 years or so, we have not been able to detect any marine pathogen in our cysts. That being said, if you do all of those things correctly, there is still an issue of the actual hatching process in the hatchery, because a lot of the pathogens that we see in aquaculture are in the environment. And so what happens during the hatching process, I believe, is that a lot of organic matter is being released, and that creates a great environment for some of these R strategist type of bacteria that can then bloom and grow, and that can create a problem. The good news is that there are some new technologies today, some new coatings that are available that are actually suppress these bacteria to a level where they're not affecting our aquaculture species. And those technologies, I think, really help with those issues that we encounter in the hatcheries. With that, I hope that I've correctly answered your question.
Thank you, Thomas. I hope we could see in the future that's a concrete protocol that's controlling of biosecurity in the maybe the update, the Artemia manual. Yeah, thank you, Thomas.
Welcome.
Thank you. For the second question, then I would like if Mr. Gonzalo, could you please answer that second question?
Okay, very good that you bring this problem up. I think my first reply to your question is that you could expect minor differences, but don't forget that the mitochondria in the Artemia can have different mutations. And since the mitochondria is the energy machinery of the Artemia, okay, any mutation can have a very important functional error, let's say that, in the transfer of electrons to provide energy. So I should say that now there is very good information that different parthenogenetic types can have differences and very useful, functional, or lethal, depending on the genes affected, on the phenotype of or the fitness of the animal. So a very simple question. Okay, you can don't see difference, but you need to try. And as Gilbert said, you need to discover these genes and what is the sequence they have to see if the protein they encode is affected. So what I suggest is that we need to do the characterization of the different parthenogenetic types. I hope I clarified this. Thank you.
Thank you. Thank you, Professor.
Thank you.
Hello, Rodrigo. I think so far it's the questions from China, from Shanghai.
Okay, very well. Thank you, Professor Ling Sui. As of that, I would like also to pose a question to the audience in Shanghai. if that's possible. Could you please direct these questions to the audience there? The questions goes like that. China is harvesting a lot of Artemia biomass from the coastal salt ponds. Does that not lower the production potential of cysts at these sites? Thank you.
Yes, I think I tried to answer the questions. Indeed, China has produced a lot of Artemia biomass, especially in coastal salt works. And I think to some extent, the cyst production could be improved by reducing the biomass harvest, but it won't be that very much because the farmers, they know when they should start harvesting the biomass and when they should stop to let the Artemia produce cysts. So for example, in Bohai Bay, the biomass harvest starts from later April and early May. And then the biomass harvesters will be reduced in June and also after mid of August when the Artemia turn into the oviparous production. So in fact, the farmers are able to balance the production of the cysts and the biomass according to the profit that they can make. So there is a balance according to farmers' judgment. Yes, Rodrigo.
Thank you. Thank you, Professor Linsui. Very clarifying. And I know that we are running a little bit over time here, but it's such an interesting topic and such a good discussion going on. I would like to at least to try to address a couple of questions from our internet audience here. And one of those I would like to bring on is the one that Karen Viverica put regarding the current status of pilot Artemia production project in Africa. And he's speaking out specifically for Senegal and Kenya, but possibly other sites as well. And also there is someone else that rephrased that sent that question, asking what else is going on within the Africa continent towards Artemia development and what are the potentialities in there. So for that, I believe that our colleague Patrick Sogelos could give some perspective on that, what's going on there. Thank you, Patrick.
Yeah, thank you, Rodrigo. And good question indeed, because Africa has quite some potential. Already quite some years ago, 1985 or 86, if I remember well, there was a successful project of Artemia production in Kenya. Gilbert can explain about the follow-up because apparently since a couple of years, there is quite some activity in relaunching, in reactivating that project with a number of small salt producers in the Malindi area. But we should also refer here to the work that was done in Mozambique a number of years ago. A pity enough that it has not been discontinued, that it has not been continued. The same in South Africa. The same, and I have seen that there was also a remark from Botswana. At a certain point of time, there was also an interest to see if some of the local salt farms inland working with high salinity wells could be used for Artemia production. So to conclude, Africa definitely has potential. There are some activities ongoing, but there is the room for much more work in that continent. Gilbert, you have anything to add?
Well, no, I think that summarizes it very well, Patrick. Maybe in terms of the natural populations of Artemia, the extent to which they have been studied, that's very uneven. In South Africa, they have done a nice exercise. Also, North African countries, there's quite some literature on this. But let's say the rest of sub-Saharan Africa, there's just very scattered information on this. And indeed, probably, as Patrick was saying, the best example where people have taken up the technique of Artemia pond production in Africa is the Indian Ocean coast, a certain area of the Indian Ocean coast of Kenya. It has spilled over into Tanzania as well. Or, okay, there are independent developments in Tanzania, maybe inspired by the Kenyan exercise, but that part of the continent, there are activities ongoing, but definitely room for improvement, for expansion, etc.
Okay, thank you both. Really great to see that that's been made, that some progress can be made as well, and for other continents, and the potentialities there be assessed and move forward. I would like also to maybe one last question from our internet audience, and that's from our colleague here in Rome, Alessandro Lovatelli, which he put a question regarding the first question that Patrick did in his presentation. Will Artemia become a limiting factor in future aquaculture development? So he's pointing out that should we ask this question as we see the annual aquaculture development slowing down? Have we identified any alternative or developed an effective artificial alternative feed? I don't know if Patrick would like to answer that, or one of the other panelists, how they see this issue and how we should move forward. Thank you.
Well, maybe I can start and see who wants to join. I think we have seen over the years that the use of Artemia has become more and more efficient. Just to give you an example, in the early years, we needed 25 kilos of Artemia to produce 1 million post-larvae of pinnate shrimp. Today, we can do it with 3 kilograms. So one thing is we have learned how to bring in formulated feeds, how can be supplement, substitute, and eventually, but this is my whole career, that I heard that next year we will have a total replacement for Artemia. I think Artemia is such a unique source of live food as starter food, but that we should try to use it as efficient as possible. So with further expansion of aquaculture, okay, there might be need for more Artemia, but there might also be more opportunities to come with earlier replacements in the hatchery development.
Thank you. I don't know if any other panelists would like to address that issue, or that's, I think it's already answered as best as possible. And so if not, I would like to also to, before I move on to the closing of this meeting, I would like to ask Simon to put out our poll in order to the participants give a quick answer for the poll itself. It's very important for us. Thank you. Simon?
Okay, thank you, Rodrigo. Our next item of business is a short poll. Of course, participation is optional, but we would really appreciate your feedback to help us understand who is participating so that we can design better webinars and programs. I'll leave the poll on screen for about 2 minutes and then we will continue. So please just select your answers and click submit when you're done. Okay, launching it now. Okay, one more minute. Okay, a few more seconds and then I'll close the poll and we'll move on. So if you can just please finalize your answers and press submit, we'd appreciate that. Okay, slowing down now, so I'll stop it. Thank you very much for your feedback. Okay. All right, so okay. Okay, Rodrigo, can you please go ahead?
Okay, thank you. Thank you all for this participation in this poll. Thank you, Simon, for putting that up. And we'll have that in the report for this meeting, of course, and it'll give a good guidance on the future actions that we might need to pursue. With that, and not wanting to overload our audience there in Shanghai, which is already a little bit past due for bedtime. But, and also bear in mind that the next 2 days we have a good program for the next meetings there. So with that, I would like to introduce our colleague, Matthias Hallward from FAO. As he's also the FAO's Subcommittee of Aquaculture Secretariat. And he's going to give us a closing address for this. And so if you please, Matthias, you have the floor.
Thank you, Rodrigo, and colleagues, friends, ladies and gentlemen, some of you, as Rodrigo said, very late evening at Shanghai Ocean University, many of you attending via video conferencing, a very warm greeting from Rome. And congratulations to all the speakers. It's my pleasure to provide closing remarks at the end of this very interesting workshop. So who would have ever thought that this small crustacean would play such a vital role in the farming of so many species of fish and crustaceans on all continents? Well, actually one person would, and of course he's among us, because history has it that it was the young Patrick Sorgelos at the 1976 Kyoto conference who at the time dared to challenge the conference chair, the late Dr. Ramu Pillai, that indeed there could be a future for Artemia use in world aquaculture, although at that time Artemia was only available from 2 sources in the US. Well, FAO took up the challenge and provided the means to verify the formulated hypothesis at the time to make Artemia a widely accessible and most suitable live food for the fish and shrimp hatchery developments that were just taking off at that time. Over the past decades, Artemia developments in availability of cyst sources and in optimizing its use in the hatcheries have contributed to the continued success and expansion of the fish and crustacean industries. However, we also heard that there's room for significant improvement, especially when it comes to the routine use of Artemia cysts in the hatcheries, where often basic principles and guidelines are overlooked or neglected. FAO has recently taken the initiative to prepare an updated Artemia manual to ensure a more sustainable and biosecure use of this important food source. We also heard that we need to worry about the future at a time of impacts of climate change on some of the vital natural resources of Artemia in many inland salt lakes. It is crucial to invest more in the study of Artemia biodiversity and the sustainable exploitation of these terminal salt lakes. In parallel, more opportunities need to be explored for controlled pond farming of Artemia in artisanal salt works, in regions with salt-affected soils unsuitable for classical agriculture, even in alluvial areas in the desert. From the examples we have seen, these new farming practices can have significant socioeconomic benefits and can be real catalyzers for new aquaculture developments. Furthermore, we should give more attention to the use of Artemia biomass as a top quality food source, not just for application in aquaculture, but as a very sustainable feed ingredient produced low in the food chain, as an extractive species, meeting all the SDG criteria. Remember the reference made in one of the presentations to the Artemia omelette consumed in the salt farms in Vietnam, which I have yet to try myself. Back in April 2018, it was at Tianjin University of Science and Technology when I had the pleasure and honor to be part of the launching of the Asian Regional Artemia Reference Center. And this was a direct consequence of a recommendation coming out of an FAO expert workshop back in 2016. I'm proud to see how our joint efforts have led to today's workshop. And it is good to learn that now, with the recent initiative to set up the International Artemia Aquaculture Consortium, there is a renewed interest for international cooperation on Artemia matters by partners from the academic world as well as the industry. A concerted and interdisciplinary effort will be needed to tackle the challenges listed, to explore the new opportunities, and maybe to consider a new leverage in Artemia development now that one has access to the Artemia genome. Finally, considering the excellent work presented here, FAO's subcommittee on aquaculture will be informed of this progress for member countries to consider further the work on Artemia within the broader context of a meaningful future collaboration towards more sustainable and more inclusive aquaculture. Thank you and stay safe, everyone.
Thank you, Matthias. And with that, I would like to get the word back to our floor there at Shanghai with Professor Min Zhang for last considerations. If you please, Professor Lin Chuan.
So, hello everyone, our friends. At the closing part of this workshop, please give me the honor to express my sincere thanks to all of our speakers and participants who attend both online and in person. It's because of your support and efforts this workshop went smooth and successful. It's also a very good beginning for the Global Conference on Agriculture Millennium Plus 20, which will be held tomorrow. Hope to see you in GCA online or offline, and mostly hope to see you in person in the future in Shanghai, China. Thank you.
Thank you for this closing remarks. And with that, I believe we can close this meeting. Thank you all for the attendance and stay safe, and we'll see you tomorrow, hopefully. Bye-bye. Good night. Good
Bye-bye.
afternoon.
Bye-bye.
Thank you, everybody.
Bye.
So recordings of the presentations will be made available on YouTube next week, and we will try to arrange to publish them on a Chinese platform as well. I'll send everyone an email with the details once they're available. Lastly, I'd like to invite you all to join the main conference, which starts tomorrow. I will leave a link to the program and the registration on screen for a couple of minutes before shutting down. Thank you and good evening.